Good morning to all of you. Actually, what we are trying to emphasize on the fact that till date, what we understand from different people, from different colleagues, that basically the principle of PCR, that is the polymerase chain reaction, is actually the amplification of the genes of interest or amplification of the region of the DNA of interest. But please make sure this is not the principle. This is basically the aim of PCR, the objective of PCR. But the principle of the PCR lies in the fact that only the tag polymerase or the DNA polymerase which you are using in your experiments can only bind to a specific double-stranded DNA. Now, what actually it means? It means when in the first step of denaturation, you denatured your DNA, then in the second step, when the annealing process happened, then your single-stranded, hypothetical single-stranded DNA will become double-stranded at specific positions where basically your primers bind from 5 to 3' regions. So now, because of these binding of primer or hybridization of primers or annealing of primer, your single-stranded DNA at these specific points have become the double-stranded DNA. So only these regions of the DNA where the primer are binding are recognized by your DNA polymerase. So once the DNA polymerase bind to this double-stranded region, the amplification process moves on towards the elongation process. So please keep in mind, this is one of the mistakes which we normally use and we normally follow when we consider the amplification or the principle of the PCR vice versa with each other. Thank you so much. Wait for some more videos in the coming future.